Good day everyone, I am Michael, the moderator for this webinar. I welcome everyone for today's webinar, which is GCP, GLP, GMP comparison and understanding of three of the FDA's major regulations. I would now like to introduce our presenter for today, John E. Lincoln. John E. Lincoln is the principal of J. E. Lincoln and Associates LLC. I would now like to hand over the floor to the speaker. Over to you, John.
Thank you, Michael, and thank you all for attending. We're going to be talking about good clinical practices, good lab practices, good manufacturing practices. The focus will be on pharmaceuticals. And so what we focus on, you can readily translate to others. There are specific FDA guidance documents that deal with devices, but the majority of them and the detail is more on the pharmaceutical side, hence the focus on that particular side, especially with clinicals. This is the agenda basically. We'll start out with the GMPs for drugs because really even when you're making samples for clinicals, you're going to be doing them under the GMPs, good manufacturing practices. Usually you see CGMP, current good manufacturing practices. Good manufacturing practices are the rock bottom, the printed legal requirements, and that you have to add to those what is your industry doing as well in general. Not leading edge, not bleeding edge, but right in the middle of the pack. Then we're talking about good laboratory practices, and we'll focus on part 58, and we'll give an example of some of the elements in a GLP compliant laboratory. And then we'll sum up with the good clinical practices, focusing on ICH E6, which is a international standard, both in the US and worldwide. Although the US has additional specifics for clinical practices, they follow pretty much the basics in ICH E6, or ICH E6 follows pretty much the basics in other clinical requirements in Europe and in the United States. We'll talk about some of the type of documents that are necessary before initiating a trial, have to be monitored or maintained and submitted during a trial, and some of the final information and some of the basic documents. We'll talk about the principles involved in clinical trials, the sponsor, the institutional review boards at each of the clinical sites where a clinical is being held, and ethical requirements of such. We'll talk about the investigator responsibilities, the key document, the protocol itself, the running of the clinical, the investigator's brochure, which is a fairly extensive discussion of the investigator's responsibilities regarding checking things, recording things, forwarding information, and to whom they are forwarded as well, and other requirements as well as periodic audits of the operation. Now, the sponsor is usually a pharmaceutical company. It could be a clinic as well, could be a group of doctors, but often it's a pharmaceutical company for pharmaceuticals or a manufacturing company for devices. So let's start out with the GMPs. We're going to focus on 21 CFR 211. 210 is just kind of an introduction. 211 gets into the basic principles. So 210 basically says here, the regulations under 210, 211 contain the minimum current good manufacturing practices for methods to be used. Now here's the key points. We have two slides here emphasizing the key subparts of the GMPs for drugs. And of course, holding has to do with the environment. The GMPs are the law. That's what's published by the Congress under the FDA's guidance. That's just some of the points involved with SOPs. Now we want to talk about the GLPs. This is 21 Code of Federal Regulation 58 primarily. And this gets into some of the basic principles for running your lab. Now dealing with clinicals, for example, you're going to be a test system. There we get into that term for animals as well as plants, etc. We mentioned the quality assurance unit. Now with storage containers, the goal usually is to replicate the original container with any supplemental storage containers you utilize as well. Here's a list of the table of contents to give you an idea what they focus on. There's subpart J, records and reports. So specifically looking at personnel, you notice here a discussion on the appropriate clothing. Now, before you start a study, you designate a study director. Your SOP should spell that out. The facilities, we've talked a lot about that, but your lab facilities is what we're focused on here. Equipment, of course, we're talking about all kinds of equipment here. We're talking about production equipment, but we're focused on lab equipment and or test equipment. Facilities, operations, and changing. Reagents and solutions need to be properly labeled. Test and control articles, we mentioned earlier, the need to separate the two. Test and control article handling. The protocol is that portion of the study that actually gets into the nitty-gritty of how you're running the tests. You notice here a description of the elements used, solvents used, emulsifiers, and these should be identified by part number and lot number. Conduct of the study shall be in harmony with the protocol, talking about the test systems. Remember, that could be animals. Reporting study results. These are in requirement regarding the clinical requirements that have usually been cleared through an agency and or certainly your IRB. Names of the individuals involved. Now, you're required to maintain these records for a period of time, so you must have a way of storing them and retrieving them. Now, the FDA requires at least 2 years following the date on which an application for research or permit. Retention is under the responsibility of the quality assurance unit. Usually they have an area called document control. We're not going to get involved in the disqualification of a testing facility. Just be aware that there are requirements for it. Compliant GLP laboratory. Suitable size and construction. That's very subjective. It is up to you to define that. Equipment, we talked about that. This gets into just the general requirements of maintaining them in a way that they're suitable for their work. You need standard operating procedures. Sometimes they're called business operating procedures, but usually SOPs, standard operating procedures. Test controllers, they need to be adequately defined. Protocol and conduct. Study should have a written protocol. The study director adheres to that as well. If you're using DOE, design of experiments for experimental design, type and frequency would be defined, records that to be maintained. Your protocol should probably reference the applicable SOPs. A final report shall be prepared for each non-clinical laboratory study and shall include the name and address of the facility. Stability would be defined, recording transformations, calculations, operations on the data. Corrections and additions, usually in the form of an amendment, other than maybe 1-2 word corrections. And where you're retaining it elsewhere, you want to have clear indication of where this information is being retained. The quality assurance unit is responsible for a lot of the retaining of records regarding this kind of documentation. Personnel, they need to appoint a study director, or even if there's construction next door to the facility, even though the study director's responsibility, they're responsible for adherence to the GLPs, etc. If you use a principal investigator, they get out of the nitty-gritty. The quality assurance unit is responsible to make sure that all the things necessary to accomplish this, facilities, equipment, personnel, methods, practices. Separateness of quality from production is important. They ensure that no deviations occurred, or if it did occur, they're adequately documented and challenged. And of course, the ability to file and retrieve, inspect the facilities at an adequate interval. Stability testing is addressed here, and stability is an important quality because and these vary depending on what products you're dealing with. Now, obviously, what you test needs to be representative of the overall. Now, whenever you replicate the study, bear in mind if you find, it's here, accelerated storage conditions, intermediate storage conditions, and good clinical practices, ICH E6, this gets into the actual running of the clinical trials. It's based on the Helsinki Agreement, electronic records and electronic signatures. This should be defined by SOP and all defined by SOP. So that concludes my discussion. Well, thank you, Michael. Once again, thank you for your attendance.
Once again, thank you, John, for the great presentations. And that does conclude the session for today. Thank you, everyone, for participating. You may all disconnect now. Have a great day ahead. Bye-bye.